नमस्कार। आप सभी लोग सकुशल अपने घर पे और डॉक्टरों द्वारा बताए सभी इंस्ट्रक्शन का फॉलो करते हुए अपने आप को सकुशल रख रहे होंगे तथा कोरोना वायरस से अपना तथा अपनों का भी ध्यान रख रहे होंगे। In previous topic we have talked about the usefulness of the microbes in household products, in which we have talked about that how the microorganisms can be utilized for the preparation of the curd, for the dough, for the 2D and the different kinds of the cheese. But later on the utilizations of these microorganisms is also used in some industrial products. Basically means they can be utilized to convert, they will act like a fermenter, right? Means they can be put down into a different large vessels where they can be make the some substances fermented, so they will act like a fermentators. So basically if we talk about the industrial products, there are two types of products are generally found. Number one is fermented beverages, antibiotics, some chemicals, some organic acids, alcohols, enzymes and bioactive molecules. Means different types of industrial products can be obtained with the help of such type of organisms. So first we talk about the beverages. First of all, you know what are beverages. Beverages means some different types of substances which can be prepared in industries with the fermenting of some fruit juices or cereal substances or malted cereal substances. Means the different kinds of the crop productions which can be fermented with presence of some microbes. Especially there are special kinds of microbes that is called fungi, especially Saccharomyces cerevisiae, which you know that it is also known as the baker's yeast or the beverage yeast. Means it can be always help to produce the ethanol with the help of different kinds of substances which can be obtained from the fruit juices as well as with the help of the different other substances in our conditions. So we can say they can be acts like a beverage. Means and these beverages can be developed with the help of this Saccharomyces cerevisiae by specific conditions. Means whenever alcoholic drinks can be developed, alcoholic drinks can be manufactured, in that conditions we have to need some raw material from the fruit juices and some other substances of the plant and plant products, as well as basically we will get the some specific type of substance which can be fermented with the help of that beverage yeast, right? In which conditions the first thing that can be obtained, they are known as that wine and beer. How this wine and beer can be developed? Means the fermentation of the fruit juices can be occurs, but this substance wine and beer can be produced without distillation. Means there is no distillation process will take place for the formation of this type of wine and beer. Means they can be always obtained with the help of without fermentations. So we can say that they can be obtained. But on the other hand, there is a whiskey, brandy and rum. They can be obtained by the fermentation of the fruit juices and different kinds of cereals, right? So they can always obtain after, or they can be formed with the help of distillation. Distillation is a process in which we can get the evaporate materials, and after condensation of that evaporating that vapor in and other vessels, so that can be known as distilled substances, right? For this generally there are different kinds of equipments, you can just see in the diagrams over there. So these are the different conditions where we can say this can be always helpful for us. Means these are the different kinds of just fermentators, and they can also help to get the distillation process over here. So again you should rememberize that wine and beer can be produced without distillations, and whiskey, brandy and rum and some other type of drinks can be always obtained with the help of the distillation. So we can get all such type of things. Then another kinds of the product is called antibiotics. Antibiotics means they can be always acts like a good source that there is no effects of some other specific pathogen on our body, right? If we talk about the this antibiotics, we can say they are the different kinds of chemical substances which can be produced by some microorganisms which can capable to kill the disease causing microbes. Means these antibiotics are the specific type of chemicals, means which can capable to kill the some special type of the microbes which can cause the diseases in our body, or they can also retard the growth of those disease causing bacteria, virus and other microbes. So these can be utilized, but basically antibiotic will not act against the viral diseases, as you have learned in the previous classes, especially that this bacteria viral does not have its outer covering, right? Because generally these antibiotics and acts on the cell wall of the microorganisms, so they can kill, but viral have the same kinds of the proteins of that organism, that's why they are not react over with that viral diseases. So generally the viral disease is not act on the such type of diseases. Okay, so as you know, the first antibiotic, that is penicillin. You all have heard about this penicillin in previous classes, but this was discovered by Mr. Alexander Fleming, and it is the first antibiotic which can be developed in the world, right? That's why we can say the Alexander Fleming was discovered with the help of. There is a very long story about the discovery of this penicillin. Means Alexander Fleming was working with a bacteria that is called Staphylococcus or Staphylococci, right? Means he is working with that bacteria, but he seen in his petri dish that where the special type of mold or yeast is grown, that is called the Penicillium notatum, there is no effects of that bacteria. Means this bacteria is not growing in that particular place where such type of mold is growing over there. So he just think that it means that this yeast will just degrade the effects of the bacteria. So this way he discovered the penicillin with the help of the Penicillium notatum. Means generally penicillin can be obtained, can be extracted from a special type of fungi that is called Penicillium notatum. But later on, basically the Ernest Chain and Howard Pollery awarded Nobel Prize in 1945 for the establishing penicillin as the effective antibiotic. Generally, penicillin was discovered by Mr. Alexander Fleming, but it is utilized as antibiotics by the other scientists. His name is Ernest Chain and the Howard Pollery, when they get the Nobel Prize in 1945 for the such type of utilizing of antibiotics, right? These antibiotics can always helpful for the functioning with the different kinds of the diseases. Means they can be cured by disease, special type of antibiotics. The some diseases like plague, whooping cough, diphtheria and leprosy, these can be always can be cured with the help of such type of antibiotics. Means in the we talk about the plague in ancient time before this corona, plague is also such type of pandemic diseases in ancient period. Means as the whole world is just suffering with this corona diseases, in the same way plague can also affect the different countries in ancient time. So these antibiotics can always act against this type of diseases, as well as generally the whooping cough, generally in our community known as kali khasi, diphtheria, that is known as galghotu, it is a type of diseases, and leprosy, everybody know, that is called the kushth rog. Means these can be capable to affect our body directly or indirectly very much. Then there is another substances or chemicals which can be obtained with the help of, that is called the chemicals. Chemicals, in case of chemicals we can say the citric acid can obtain by an special fungus that is called Aspergillus niger. Means we can get the product as citric acid with the help of a fungus that is called Aspergillus niger. On the other hand, the acetic acid can be also obtained with the help of a bacteria that is called Acetobacter aceti. Means this acetic acid can be always obtained with the help of the Acetobacter aceti. It is a bacteria, right? And the butyric acid can also obtain with the help of a bacteria that is called the Clostridium botulicum. Means butyric acid always obtained with the help of Clostridium botulicum bacteria. And lactic acid, you can get lactic acid with the help of Lactobacillus. Means it is a bacteria that is called lactic acid bacteria. Means which is generally utilized for the converting of the milk into the curd, so they can be utilized. As well as the ethanol can be also produced and obtained with the help of the yeast that is called Saccharomyces cerevisiae, that is called beverage yeast or the baker's yeast. It can be also helpful to obtain such type of ethanol to us. Then we have to talk about that there is another thing that is called the enzymes. Means there are some enzymes. What are enzymes? Enzymes generally you can say they are the catalyst which can always helpful for the increasing or reducing the different kinds of chemical process in our daily life. As well as these enzymes can also utilize or develop in the industry for different purposes. If we talk about the some special kinds of enzymes which are given in our syllabus, that is lipase. Means lipase, as you heard this lipase enzyme in your digestive system in previous classes, where you know that it can always helpful for the digestions of the lipids and the fats. But here this lipase can be produced by the industry for the formations of the detergents, right? Because it can be used in the detergents formulations. It can be always used for the making the detergents, which can be always helpful. This lipase enzyme called always helpful to remove the oil stains from the laundry. Means generally, as you know, whenever there are lots of patches can be found or due to any some oily patches on our clothes, they can be removed only with the help of this lipase enzyme. If we talk about some other, that is called pectinase or protease enzyme, they can be also useful for that pectinase and protease enzyme. They can be used to especially for the clearing the bottle of juices. As we all know that we get the different kinds of the juices in the bottles, but if they should be not cleaned with the help of such type of enzymes, they can be damage the preservative material which are preserved inside those bottles. So in the industry, basically this pectinase and protease enzyme can be utilized for the cleaning of the different kinds of the bottles or of the juices. And on the other hand, there is some special type of enzyme that is called streptokinase. I think everybody know about this streptokinase. It is very important. So many times it can be asked in our syllabus or in NCERT questions as well as in board questions. So this streptokinase can be obtained from the streptococcus bacteria. There is a bacteria that is called streptococcus, which can be always very helpful. And you know that this streptococcus bacteria can produce this enzyme by the modified with the help of some specific type of biotechnology. Means on the basis of the genetic engineering it can be modified, which can be utilized in different places. So you can say especially this streptokinase acts like a clot busters. What do you mean by this clot busters? As you know that in our blood vessels, right, blood can be get clotted, or they can sorry they can make some special type of clots. So we have to use this streptokinase enzyme to break those clots from the blood vessels or of any patient. So it can be always help for us. Means it can always helpful for the myocardial infections persons. So streptokinase is very important enzyme for the difference. Sometimes these enzymes can be asked in question paper, so you must try to learn their functions and their names also. Okay. After that, there is another type of molecules that is called bioactive molecules. First of all, you must know what are bioactive molecules, how they can be always helpful for the different organisms. Basically, these bioactive molecules can also prepare with the help of industry. Means we are talking about the microbes utilized in industry products. Means how the microbes can helpful for the production of such substance, in which we are talking about the bioactive molecules. There are especially two bioactive molecules are given in our syllabus, but there are lots more than these can be also found in everywhere. So we can say, if we talk about the cyclosporin A, it is a bioactive molecules that is called cyclosporin A and stannine. So how this cyclosporin A can be obtained? Cyclosporin A can be obtained with the help of a fungi or fungus that is called Trichoderma polysporum. Means it can be always get or obtained with the help of a fungi that is called Trichoderma polysporum. And this cyclosporin A can be utilized or used especially for the immunosuppressive agent in organ transplant patient. Basically it can be always helpful for during or utilized during the immunosuppressive agents, means for the organ transplant patient. Means whenever there is an organ transplant can be from one person to another person, in that condition they can be utilized over there. That's why we can say it is very important biomolecules, bioactive molecules. So they can be utilized in all the places, and it can always help for the immunosuppressive agents. Means because in that particular time when the organ transplant will take place, in that particular time it is very needed by the in such conditions, because this it can acts like an agent for the organ transplant patient. It acts by the inhibiting the activation of T cells. Means this cyclosporin can always inhibit the activation of T cells. Suppose A person organ can transfer to B person, right? In that particular conditions, his cell immunity, right, can affect at that particular time. So this cyclosporin can suppress immunosuppressive agents during that conditions. So this cyclosporin can act like a inhibiting the activation of T cells of that patient. So the T cells will not actively participate in that particular condition, so they can be easily, organ can be transparent and the organ can be work properly in that patient conditions. And the second bioactive molecule that is called stannins, right? This stannins can be obtained or developed with the help of a yeast that is called Monoscus purpureus, right? Monoscus purpureus yeast. Basically this can be used for the clot busters, or sorry, not clot busters, it can be always used for the lowering the cholesterol in the human body. Means this can be always helpful for the lowering of the cholesterol in the patient. So they can be acts especially for the blood cholesterol lowering agents. Means statin can always help for the lowering of the blood cholesterols, because they can act by the competitively inhibiting the enzymes responsible for the synthesis of the cholesterol, because this stannin can always help to inhibit the function of the different other enzymes which can capable to increase the cholesterol level in the blood. So we can say, just like they can be always against, or they can always oppose against some specific type of enzyme that is called paravastatin or the simvastatin. Means simvastatin and paravastatin. This simvastatin and paravastatin can always help for the increasing or synthesizing the cholesterol level in the human body. That's why we can say this statin can always helpful for lowering the cholesterol level in the body. So this way this topic can also completed. So please quick revision for that, this how these microorganisms can helpful for the industry products, for especially if we talk about the production of the citric acid, there is a fungi that is called Aspergillus niger which can always help for the development of this citric acid. On the and acetic acid can be developed with the help of Acetobacter aceti, and the butyric acid which can be also very important for developing with the help of a bacteria that is called Clostridium botulinum, as well as lactic acid which for the utilization of the curding that can be obtained with the lactobacillus, and ethanol can be always developed with the help of the yeast that is called Saccharomyces cerevisiae, and different enzymes like lipase which can help for the removing the oil stains from the clothes or laundry. It can be used for the making the detergent, as well as pectinase and protease enzymes which can always help for the clarifying the juice bottles and cleaning for the cleaning of the juice bottles. As well as if we talk about the streptokinase enzyme can always use obtained with the help of streptococcus bacteria and use or acts like a clot busters for removing the clot from the blood vessels of the patients, because if there is any clot can be obtained, so the patient can be affects a disease like the heart attack. And the last that is called stannin, which can be always obtained from the yeast that is called Monachus purpureus, have been commonly used to for the lowering of the blood cholesterol agents. So it can be also utilized over there, and these stannin can also acts or inhibits the synthesis of cholesterol against the enzymes like paravastatin and the simvastatin. So students, you must completed this topic, and later we will talk about another topic of this chapter which is also very important, but you all must prepare the notes of these lessons in your homes. Thank you.